Hello everyone, my name is fady and this course under supervision of Dr. and Prof. Hussein Sabit and Dr. and Prof. Hala Isa. Today I will talk you about the commonest monogenic neurological disorder in the developed world, Huntington disease. This disease first described in detail by George Huntington in 1872. There are about 10.6 to 13.7 individuals per 100,000 are affected in Western populations, and the prevalence of the disease is lower in Chinese and Black African populations. And among the Japanese, the prevalence is estimated at below 0.38 cases per 100,000 population. So they are my outlines. Firstly, what is Huntington disease? Then Huntington structure and function. Then clinical description of Huntington disease. Then development of the disease and diagnosis of the disease and management and treatment of the disease and genetic approaches to Huntington disease. Okay, let me talk you about genetics of Huntington disease. In 1993, the gene associated with HD was identified, the HTT gene, located on chromosome 4b 16.3, contains a variably expanded CAG trinucleotide repeat sequence in exon 1. The normal allele contains a repeat sequence of 10 to 35 CAG, and triplets encoding a polyglutamine tract proximal to the N-terminus of the protein Huntington, and expansion of the CAG repeat tract beyond this range is associated with the development of HD and is transmitted through autosomal dominant inheritance. Huntington structure and transformations. Expression of HTT generates an initial RNA transcript that's normally processed into an mRNA encoding the full-length Huntington protein, but it can also be apparently processed into a mRNA encoding only exon 1 if the gene contains an expanded CAG repeat. Translation generates either the full-length Huntington protein or the HTT exon protein. The HTT exon 1 fragment consists of the 17 amino acid mixed sequence HTT-NT. The polyglutamine polyQ sequence encoded by the CAG repeat and a proline-rich domain PRD. The full-length Huntington protein consists of this exon 1 sequence followed by a series of ordered and disordered protein segments. Proteolytic cleavage. As label 5, cleavage sites indicated by arrows, mediated by recognition sequence. Located in the disordered segments generates a series of products including HTT exon 1 like fragments. Such fragments containing expanded polyQ segments have important role in triggering Huntington disease by molecular mechanisms that are yet to be elucidated. Clinical description of Huntington disease divided to: first, motor disorder like difficulty with speech and chorea and dystonia and impaired gait or balance. Second, cognitive disorder. Cognitive impairments often associated with Huntington disease include difficulty organizing and focusing on tasks, difficulty in learning new information, and lack of awareness of one's own behaviors and abilities, and slowness in processing or finding words. And third, psychiatric disorder. Psychiatric disorders like feeling and sadness or apathy and frequent thoughts of this and fatigue and loss of energy and insomnia. This table shows development of the disease. Development of the disease divided to two mainly stages. First, called preclinical stage, and second, called clinical stage. In preclinical stage contains three stages. First, at risk stage 50-100% one affected parent, and second, gene carrier pre-manifest stage, and third, transition phase. And in clinical stage contains also three stages, called first clinical stage 1 and clinical stage 2 and clinical stage 3. Diagnosis of the disease done by five things. First, neurological examination like motor symptoms, sensory symptoms, psychiatric symptoms. Second, neuropsychological testing to test the memory and reasoning and language skills. Third, psychiatric evaluation like coping skills and emotional state and patterns of behaviors. Fourth, brain imaging and function by MRI or CT scans. And fifth, genetic counseling and testing. Management and treatment of the disease. There is currently no effective treatment available to stop the progression of Huntington disease, but medications can decrease some symptoms of movement and psychiatric disorder, like medications for a movement disorder and medications for psychiatric disorders. Genetic approaches to Huntington disease divided to DNA approaches and RNA targeted therapies. DNA approaches like transcription activator like effectors, and RNA targeted therapies like RNA interference like small interfering RNA and short hairpin RNA and microRNA, and antisense oligonucleotides. Finally, conclusion. Huntington disease is a destructive disease caused by a genetic mutation that's readily identified independent of the onset of disease symptoms. However, understanding the molecular and cellular pathogenic mechanisms that underlie the development of disease phenotypes remains a significant challenge. The importance of specifically targeting MHTT, optimal drug distribution within the brain and in the periphery, and the spectrum of target and other side effects. It will be essential to perform studies in children whose phenotype differs significantly from that seen in adult onset disease. And thank you.